Let us look into FDA-approved clinical nano drug delivery systems. Currently, passive targeting is used by the majority of FDA-approved nano drug delivery systems. However, a lot of active targeting nano drug delivery systems are waiting for FDA approval. Therefore, they will soon be available in medical applications. Several nano drug systems have been developed and FDA approved for clinical uses. A large number of nano drug systems are also awaiting FDA approval. Liposomes, micelles, dendrimers, and polymeric nanoparticles are used as drug carriers in nano drug system. The cancer drug is either inserted inside or bonded to the carrier structural surface. There are few clinical nano drug systems that uses active targeting in drug delivery to the tumor location until now. Passive targeting is used in most of current nano drug systems that are used in clinical trials to target cancer tissues. DPR effects cause more nano drug systems to be selectively observed by the tumor location. The pH or temperature sensitive nano drug systems interact with the cancer tissues after being observed by the cancer tissue and then are broken inside the cancer tissues to release the drug. Many active targeting nano drug systems are undergoing extensive development and are expected to be employed in clinical trials soon. This table shows the nano drugs which were approved by FDA. Nano carriers, liposome, micelle, protein, and lipid. Okay. And this nano drug system are used for various diseases. And this table shows preclinical active targeting nano drug delivery system. On top is only one approved by the FDA in 1999, and other drug delivery systems are waiting for FDA approval. That's phase 1-2, phase 1, preclinical, and most of the nano drug system using liposome for nano carrier or polymer or colloid, and the targeting receptor as protein, IL-2 protein, transferrin. So this targeting transferrin, and the GAH antibody. Okay, and this drug is targeting breast cancer receptor as galactose. Okay, so and the tumor as liver cancer or solid cancer for these drugs, leukemia or some other stomach cancer, and several other diseases. So also liposome as nanocarrier, so as polymer, and targeting receptor as enzyme, this one, and anti-EGFR. Okay. Now let's see the FDA approved nanodrug system in details. First, let's see the Doxil. Doxil is the first FDA approved nanodrug system in 1995. Doxil employed pegylated nanoliposomes as carrier, which extended the drug's circulation period and prevented the drug system from being captured by the RES. Because of the EPR effect at the tumor cells, Doxil is passively targeted to tumors, and its doxorubicin, this cancer drug, is released inside tumor tissues. This doxorubicin cancer drug can be used to treat several cancers, including breast cancer, breast cancer, Kaposi sarcoma. This is the disease, the malignant tumor on blood vessel, lymphoma, and acute lymphoblastic leukemia. So this doxorubicin, this cancer drug is used for several cancers. The FDA allowed the cancer drug doxorubicin approval to be used in 1974. Okay, the nanocarrier is liposome, and inside the center of the liposome, doxorubicin is placed, and outside coated with the PEG, so pegylated. Then this PEG actually make the drug's circulation period extended, and also prevented the drug system from being captured by RES. Next, let's see the Abraxane. In this Abraxane, the cancer drug is paclitaxel. So paclitaxel as cancer drug is stabilized inside albumin in the nanodrug system in Abraxane. So this albumin cell, so inside albumin cell, cancer drug paclitaxel is placed. The overall size is about 130 nanometer. The size 130 nanometer. The schematic structure of Abraxane is shown in the figure. This figure. Paclitaxel, which is cancer drug, is implanted inside nanoparticles. Albumin binds to hydrophobic molecules with high affinity. As a result, it's an excellent vehicle for delivering water-insoluble drugs to tumors. So paclitaxel is water-insoluble drugs. The cancer drug paclitaxel is effective against ovarian, esophageal, breast, lung, Kaposi sarcoma, cervical, and pancreatic cancers. In 1971, paclitaxel was first extracted from Pacific Eel, and in 1993, it received approval as cancer treatment. Okay. And now let's see a DP docetaxel. The drug carrier in DP docetaxel is dendrimer structure. So that's dendrimer structure. Each molecule of docetaxel is attached to the dendrimer scaffold. The linker between the docetaxel and the dendrimer is intended to break in the cancer site. So this is cancer drug docetaxel is attached to the branches of dendrimer. So the link between a docetaxel and dendrimer is intended to break in the cancer site and there releasing the docetaxel cancer drug into the cancer site. Docetaxel, which is cancer drug, is made from Texas tree. Docetaxel interferes with the microtubule separation mechanism, which stops the proliferation of cancer tissues. One of the celled organelles, the microtubule, is crucial to the process of cell self-replicating. So this docetaxel, the cancer drug, actually stops the proliferation of the cancer tissue, and then the cancer tissue will be eliminated. The bond between docetaxel and the dendrimer branch in the nanodrug system is dissolved once the drug has reached the tumor tissues, allowing the drug to be released into tumor tissues. So, in the cancer tissue, the bonding between the docetaxel and the branch of the dendrimer is broken, and docetaxel, the cancer drug, is released to the cancer tissues. Now, olivite Onivide makes use of the liposome structure to store the cancer drug irinotecan. So cancer drug is irinotecan. The surface is coated with a PEG. So surface is coated with PEG. In 2015, Onivide received the clinical approval by FDA. Irinotecan, a cancer drug, is a topoisomerase retrainer that prevents a certain enzyme from being used in DNA replication and cell division, which eliminates cancer tissues. So irinotecan is put inside the liposome, and the liposome is coated with the PEG outside. Okay, so lipid membrane is here. The production of the protein is stopped if a short piece of synthetic RNA is attached to the mRNA. This phenomena known as RNA interference. The discovery of this phenomenon was made in 1998 by Andrew Bayer. So we can control the expression of specific gene by using this RNA interference. However, we had to figure out a secure way to deliver the short RNA piece to the target organ for non-drug system that uses RNA interference. So RNA was supposed to reach the target organ throughout this delivery process without dissolving in the blood vessel and was subsequently released by the kidney. By encasing the RNA molecule inside lipid nanoparticles on patro, this nanodrug system, was able to securely deliver the drug to the target organ. This nanodrug system has been approved for use in the treatment of hereditary transthyretin amyloidosis associated with polyneuropathy. This disease is a neurodegenerative disease caused by genetic mutation in the transthyretin protein. So this Onpattro is approved for treat this disease, and the RNA is encapsulated inside the nanoparticle. This Onpattro is the first RNA interference-based nanodrug system. Was approved for clinical uses in 2018. Now let's see some of the targeting nanodrug systems. These targeting nanodrug systems are waiting for FDA approval. First one is CAA. The nanocarrier for the CAA-LA is made up of polymer nanoparticles. That's polymer nanoparticles. The targeting ligand of transferrin is attached on the surface of nanoparticles. So there is transferrin is attached to the surface of nanoparticle. The siRNA for DNA interference is inside the nanoparticles. Inside nanoparticles. This system of nanodrug targets the ribonucleotide reductase M2 RRM2 subunit. This nanodrug system inhibits the growth of tumor tissue by DNA interference. So this DNA, siRNA, inhibits the growth of tumor tissue by DNA interference. Next, this one. SIG12D loader has a cylinder-shaped polymer matrix structure made by polylacticoglycolic acid. This one has a cylindrical shape. In about 30% of cancer cases, the KRAS transition is present. This transition is prevented by KRAS G12D. So these are put inside in the cylindrical shape. Okay, so this KRAS G12D prevent the transition. So the cancer tissue will be eliminated. Now this one, MCC465. Nanocarrier of MCC465 is liposome. So liposome structure. This nanodrug system targets the monoclonal antibody GAH, which is found in the majority of cancers. The liposome acts as MCC465's nanocarrier. Okay. So targeting the GAH antibody and pair coating. The monoclonal antibody GAH, which is present in the majority of cancers, is the target of this nanodrug system. This system's drug is doxorubicin. Next, this one. The epidermal growth factor receptor, EFGR, which is overexpressed in the majority of solid tumors, is the target of this anti-EFGR-IL drug system. This system's drug is doxorubicin. The pegylated liposome acts as nanocarrier, the pair coating outside the particles. It's difficult to pronounce. phosphatidylserine, the PS, is present in the areas between the double phospholipid layers of normal tissues. However, in cancer tissues, high levels of PS are observed across the cell surface. In normal tissue, double phospholipid layers, so this PS is present in area between the double phosphorylations, so in normal tissue. However, in cancer tissues, PS are observed on the surface, cell surface, so that's different. This PS is the target of SAPC, so this system. The pancreatic, lung, brain, and pediatric cancers may be treated with this nanodrug system. This drug system is able to enter cells due to its PS bonding in the acidic environment. By mixing SAPC and the DOPS in a low pH environment, SAP DOPS vesicles are synthesized. So this is a process. So this one, SPAC and DOPS as combined, then we can have SAPC DOPS drug system. Right. This nanodrug system has an average diameter of the 200 nanometer. Okay, so next this one. The drug release process of the thermodox nanodrug system is depicted in the right figure. So this figure. So by using external ultrasonic or radio waves, the heat is produced locally at the cancer. So in this way, we can use ultrasonic or radio waves. Then heat is generated in the cancer site. The drugs inside the liposome are then released into the cancer tissues as temperature-sensitive liposome of the nanodrug system is broken. Therefore, cancer tissues are eliminated. So, in this process, thermodox nanodrug system is used to eliminate the cancer tissues.